So in this particular session, we are going to move away from the classroom style format where I'm standing at the front of the room and put the images that we're going to be talking about on the screen. This is mostly from the standpoint of it would require a lot of drawing on the board, and in this case, it's easier for me to do the same drawings on paper and then load those diagrams onto the screen for a clearer view. Now, before we get into that, however, there is one topic that was overlooked when we talked about cardiac muscle tissue, and that was the intracellular activity. Now, this was a topic that was probably covered with you when we did skeletal muscle back in A&P1. These topics, whether it's skeletal or cardiac, doesn't really differ much. It's more so in the structure of the cardiac muscle cell, not so much for the internal workings, at least not at the level that we're going to be responsible for. But when we talked about the cardiac muscle cell in one of our previous lectures, we talked about these short branching cells that linked end to end with intercalated discs being that main site of attachment for structural integrity between one cardiac muscle cell and its neighbor, but also with these embedded gap junctions. It was the gap junctions that made cardiac muscle tissue operate as a unit, so that the independent muscle cells in that tissue didn't require independent innervation. It's the gap junctions that made it possible for that one location of the SA node to depolarize, to send this wave-like manner of contraction over the heart muscle. On the surface of the sarcolemma, you can see these small holes. Those are those entrances for the transverse tubules, and they would extend down deep into the internal structure of that cardiac muscle cell, providing a connection between the sarcoplasmic reticulum deep in the cell and the sarcolemma, which is going to be transmitting that electrical conductivity. The sarcoplasmic reticulum down deep in the cell is where we have calcium released. And if you remember from our discussions of skeletal muscle, we needed calcium to be released from the sarcoplasmic reticulum so that we could make muscle contraction possible. So what we see at the SA node is that spontaneous depolarization at a regular rate and rhythm, causing the cells of the SA node to undergo an action potential. Then we see that action potential spread across the cardiac muscle tissue because of the structure of the cells with their gap junctions. So when we look at the internal structure of the cardiac muscle cell, we will see that electrical impulse run along the surface of our cardiac muscle cell, that action potential. It will use the transverse tubules to carry that electrical signal down deep into the cell. And in those transverse tubules, we get calcium voltage gated channels. Remember, calcium was one of our ions that was in higher concentration outside the cell. So if we have an action potential moving along the surface of that cardiac muscle cell down its transverse tubule, we will open up these calcium voltage gated channels, at which time the high concentration of calcium will move through those particular channels and into the cardiac muscle cell. Here is our sarcoplasmic reticulum deep in the muscle cell, containing very high concentrations of calcium ions. When our calcium enters the cardiac muscle cell, they will bind to what's called a ryanodine receptor on our sarcoplasmic reticulum. And we get a phenomena called calcium-induced calcium release. Now think about that name, calcium-induced calcium release. Calcium enters the cell and induces a massive dump of calcium from the sarcoplasmic reticulum and into the cytosol of that cardiac muscle cell. So the calcium entering the cell induces the massive release of calcium from the sarcoplasmic reticulum. Calcium-induced calcium release. And from here, we just simply continue on with our normal skeletal muscle model as it was described before. This calcium binds to troponin and pulls the regulatory proteins away from our myofilaments. Then as long as we have ATP to break down, we can undergo our sliding filament hypothesis in order to get muscle contraction. Now, if that last section is a little hazy for you and you need a review, I encourage you to look into the textbook under our muscle physiology section in the textbook, and you can get your review there. If you have any questions, please let me know. But I want to move on to heart rate regulation now. And before we do that, let's look back at a term that we also breached a long time ago called homeostasis. Homeostasis is a state of the body that is being maintained in narrow ranges, but still being able to change in order to adapt to the environment inside and outside the being. And we notice any number of variables are under what we call homeostatic control. Body temperature is one of the easiest to explain. Our average body temperature should be about 98.6. It can go a little higher, a little lower, but we have mechanisms in place so that if that body temperature gets too high, we bring it back down. Think about what happens when your body gets too warm. Sweating. We get blood vessel dilation close to the surface of the skin to dissipate heat, all in an attempt to bring that body temperature back to where it should be. If body temperature gets too low, we see different phenomena, but with the same intention, to bring body temperature back to where it should be. So if you are noticing a decrease in body temperature, now you might see skeletal muscle undergo a twitching, shivering response. You may see these same blood vessels that we mentioned a moment ago, but this time constrict, shunting more blood to the core of the body to maintain core body heat. Heart rate is one of those items that are under homeostatic control. We have mechanisms in place during such times when we need higher or lower heart rate. So we are always taking these different variables. Heart rate is just the topic of the day, so to speak. But we're always taking these different variables and detecting stresses in our internal and external environment so we can make appropriate changes internally and maintain the viability of the being. Now, generally speaking, we maintain homeostasis using two broad types of mechanisms, positive and negative feedback. Now, for us right now, we can take positive off the table. We only have one example of positive feedback, and that was going to be the birthing process. Obviously not what we're talking about now. Negative feedback is the means by which all of our other homeostatic variables are regulated. A typical feedback loop, regardless of whether we're talking about positive or negative, will always have these three generic components. We have a receptor component that's always detecting changes internally and externally. It's going to be feeding information, whatever it detects, onto a control center. And it's the control center's job to interpret and respond if necessary. So again, picture the thermoreceptors in your body, detecting body temperature, passing on that information constantly to control centers in the hypothalamus of the brain. Little changes in body temperature, 98.7, 98.8, insignificant. We are not going to be worried about responding to those sort of changes. However, as that body temperature continues to move further away from the norm, it becomes increasingly more likely that we'll have to respond and counter. So if we get too far from the norm, now our control center is going to recruit any number of effector organs to make the changes necessary. With our body temperature analogy, we just mentioned a few different effector organs. Blood vessels dilating and constricting. Skeletal muscle doing a shivering response. Glandular tissues in the integument secreting sweat onto the surface of the skin to dissipate heat. Those are all be effector organs recruited by a control center if necessary. And we get this constant circular pattern of detecting, correcting, constantly monitoring all these variables in the body and making the appropriate changes. And when we look at how that negative feedback works with something like temperature, we see it take on this sinusoidal wave. Fluctuations around the set point with an expected floor and an expected ceiling on how far we can deviate from the norm. Now, a lot of the mechanisms we're going to talk about this semester are going to involve the sympathetic and parasympathetic nervous systems. And aside from the details of what we're going to talk about here coming up, the concept on which we're going to apply next is going to be critical and a major stumbling block for a lot of students. You studied both of these divisions in A&P 1. You probably discussed the sympathetic nervous system as the half of the involuntary nervous system that amps the body up. It increases blood pressure, it increases respiration rate, pupils are going to dilate, increases or decreases gastric motility, decreases urine production, but it also increases heart rate. And we often gave it the tagline of fight or flight. Parasympathetics do the exact opposite. In this case, it's going to lower heart rate, and it's given the term rest and digest. Now, the problem with the order in which we study these topics in is that you get to look at these two divisions and think about them along the lines of sympathetics increase heart rate, parasympathetics decrease heart rate. And that's not false. But it, in a sense, puts the cart before the horse, makes you start thinking about it in the improper order, whatever the case may be. We need to step back for a moment and look at what's going on. Never picture these two entities to be the causative factor. Never think about what the sympathetics and parasympathetics do per se. Go one step back and ask yourself, why are the sympathetics or parasympathetics being recruited to carry out their intended function? Because remember, receptor, control center, effector. We are receptor-driven organisms. When we talk about sympathetic and parasympathetic acting on an end organ, this is the part of the cycle that we're mentioning. Sympathetics going to the heart to increase heart rate. Parasympathetics going to the heart to decrease heart rate. I don't want us starting here and thinking we're understanding the feedback mechanism, because we won't be. And starting from here is only going to give you problems. We have to start from here. Always. We are receptor-driven organisms. So we always have to think about what's happening at the receptor level that's resulting in the control center recruiting sympathetics or parasympathetics. They will adjust heart rate in the direction that we predicted, increase heart rate or decrease heart rate, but only because of what's being asked of them. The tissues are going to demand more or less blood. That demand is going to be detected by the receptor. Then we use the sympathetics and parasympathetics as the tools to change heart rate to where we need it to be to move enough blood to supply the tissues. So let's look at what we're talking about here now. This is our feedback diagram. This is sympathetic innervation, which we already discussed would result in a fight or flight phenomena and an increase in heart rate. But we have to ask ourselves why we need more blood. So let's say, hypothetically, you get up from your computer right now and you start going for a run. What's going to happen to oxygen levels in the blood as you start to use the muscle more? What's going to happen to carbon dioxide levels? They are going to change in the blood, and we are going to see altered blood composition from the perspective of those two particular gases. We have two different areas in the body where we're going to be concerned about right now, where these receptors are going to be located. First of all, we have a group of peripheral chemoreceptors. These peripheral chemoreceptors are going to be located in the aortic arch, aorta being the large blood vessel that leaves the heart, arches around before it dives down into the abdomen. So we have peripheral chemoreceptors in the wall of the aortic arch, and we have peripheral chemoreceptors in the walls of the carotid sinuses as part of our carotid arteries. We have central chemoreceptors in the medulla oblongata, actually in the wall of the oblongata, adjacent with the fourth ventricle that runs down through it, detecting changes in oxygen carrying capacity and carbon dioxide capacity of the blood. There's your receptors, always detecting changes to oxygenation. In this case, we see a decrease in oxygenation. We need more blood to be pumped. Our peripheral and our central chemoreceptors will now fire into a particular part of the brainstem called the nucleus tractus solitarius, or the solitary nucleus. Nucleus tractus solitarius, or the solitary nucleus. That's going to be the part of the brainstem that will be collecting information from these central and peripheral chemoreceptors. Then the nucleus tractus solitarius will recruit another portion of the medulla to either speed heart up or slow it down. In this case, we're talking about the sympathetics. We're looking at speeding the heart rate up. So we have a cardiac acceleratory center located in the medulla. The nucleus tractus solitarius sends a signal to the cardiac acceleratory center. Sympathetic nerves leave that center in the medulla and go to the SA node of the heart. From here, this is what we talked about last class. Those sympathetic nerves, norepinephrine from the sympathetic nerves, innervating the beta adrenergic receptors at the SA node, results in excess sodium entering the cells of the SA node through the HCN channels. We more quickly depolarize the membrane with our pacemaker potential, and we cut down the time it takes in between the beats, giving us more beats per minute. So we increase heart rate. We increased heart rate not just because the sympathetics told the heart to increase heart rate. We increased the heart rate because we saw changes in blood oxygenation and blood carbon dioxide levels, which resulted in our control center recruiting the sympathetics to make the proper adjustments. So we saw a need for increased blood movement to the tissues due to an increase in metabolic demand. Now, conversely, just look at any good negative feedback loop, we need to be able to make corrections in two different locations. So now you've gone for your run. You just got done running a mile or so. Your heart rate has been elevated to meet the needs that the muscles had for more blood. Now, you're done with your run, you come home and you sit down. Heart rate keeps staying elevated for a period of time. Respiration stays elevated for a period of time. But eventually, with now the muscle activity died down and the metabolic activity lessened, eventually you will start to regulate your blood gases more. Bring them back to where they should be. That normal situation now is going to be detected by the peripheral chemoreceptors in the aortic arch and the carotid sinus, as well as the central chemoreceptors in the medulla. Again, still passing on signals to the nucleus tractus solitarius. This time, because we no longer need that elevated heart rate, the nucleus tractus solitarius now recruits the cardiac inhibitory center. And by way of the cardiac inhibitory center, we are going to send a parasympathetic nervous impulse using the vagus nerve, cranial nerve 10, dumping acetylcholine onto muscarinic receptors of the cells of the SA node. Now here again, exactly what we talked about last time. Now the SA node, we still have sodium leaking in through the HCN channels, just like we did at rest, but the parasympathetics make that membrane more leaky to potassium. We get excess potassium leaving the cell, countering some of the sodium that's entering the cell, making it take longer for our pacemaker potential to depolarize, putting a little bit of extra time in between each beat, therefore lowering the beats per minute. So when you're answering questions on the exam, you have to really be particular about what that question is asking. If you get a question that asks, which of the following divisions of the nervous system increases heart rate? That's going to be sympathetics. Which of the following divisions of the nervous system decreases heart rate? That's going to be parasympathetics. However, if you get a question along the lines of heart rate is elevated, what will happen to sympathetic nerve function? Well, if heart rate's already elevated, and we are always trying to bring our heart rate back to the average, then in a case like that, sympathetic activation is going to go down because the heart rate is already elevated. It's the phrasing of the question. If you want to elevate heart rate, you need sympathetics. However, if the heart rate is already elevated, sympathetics turn off and parasympathetics are going to turn on. So please be careful with the phrasing of the question, and always be thinking about any of these feedback mechanisms that we'll be talking about moving forward from a receptor first standpoint. Always consider why and what's happening in the tissues that's resulting from this increase in activity. And if you start looking at it from that standpoint, it will make everything come together so much more clearly and quickly for you. So please review this section in particular. It's the last section we're going to have on heart rate. And if you have any questions, let me know and we'll handle it. Take care and good luck.